disclose all the questions. Thank you. Let us see the last part of these nanomaterials, polymeric nanomaterials and devices. Now, this slide shows, the title of this slide is hydrophilic, partially degradable, bioactive acrylic cements. So, look at the keywords. Is a hydrophilic property, partially degradability, that is another property, bioactiveness is another property, and this is the material. So, we can think of some material which can provide so many different properties. Now, till now you have seen these different systems, hydrophilic, hydrophobic, for making nano devices, all those things, for delivering drugs in nano dimension, all these things. Now, we are thinking of the matrix, or you are thinking of the support, we are thinking of the carrier. Okay. So, these are the things. So, a system may be hydrophilic and you have to make it degradable, partially degradable. You know the ways and means how to make a system degradable. If you take hydrocarbon, it is not degradable. If you take non-hydrocarbon, polar materials, it is degradable. So, you can make a system partially hydrocarbon type, partially non-hydrocarbon type, and you can do it. Okay. So, this way you can get partially degradable hydrophilic. So, HDBC, biodegradable composite, hydrophilic degradable bioactive composite. Okay. What are the materials we can think of? This chitin and chitosan. What is the source of chitin? Do you like lobster, prawn, crabs, shrimps? The cells of those things contain chitin. There is a naturally occurring polymer having such formula AC. These groups are there. Carbohydrate like moieties with some substitutions, acetyl groups. Now, this chitosan is a deacetylated chitin. Chitosan is a deacetylated chitin. Okay. Now, this chitosan or chitin has found extensive use in biomedical field. This polymer, natural polymer, with little bit of modification, many different kinds of biodegradable, bioactive systems are being made. I am not going to detail, just for your information. These are the formulas and structures, names are given here, you see later on. Okay. It is not difficult to understand. So, chitin is deacetylated chitosan. Okay. Applications, their toxicity is low, this is inert in the gastrointestinal tract of mammals. So, this can be used for drug delivery purpose. and this can be used for a wound dressing material, wound dressing. That means, you can make a fabric, you can draw or you can spin fiber from these chitosan molecules. You can make a fabric, that fabric can be used for wound dressing, that will be better fabric than cotton and other things. Then you can make a composite of hydroxyapatite, chitin and chitosan, which can be used for bone filling, all these things. Okay. Now, there are some modifications on this chitin and chitosan, sugars, dendrimers, cyclodextrins, crown ethers and glass beads, and those find some suitable applications. So, these are all chitosan related developments and devices which can be used for various biomedical purposes. Now, look at biodegradable polymers as biomaterials. Could you tell me one example of biodegradable polymer? PLA, you should say immediately polylactic acid, polyglycolic acid, a copolymer of polylactic acid and glycolic acid, those are natural polyesters or your biodegradable polyesters. You can synthesize this starch also, this chitin and chitosan, these are also biodegradable. So, one can use these polymers for making 3D porous scaffolds, 3-dimensional porous scaffold for tissue engineering, as well as controlled and sustained release of drug or for drug delivery purposes or drug delivery vehicles. And polylactic acid, as I mentioned, polyglycolic acid, they are copolymers, those are used in implants as sutures, as matrix materials for tissue engineering. These are examples you see. Polyglycolide, resorbable suture, commercially available. Dexon is the trade name of this suture. Fully absorbable suture, it need not removable after healing of the wound. Polylactides, suture, tissue fixation screw, scaffold for ligamented replacement, blood vessel, conduits, drug delivery vehicles. It is a copolymer, lactide and glycolide copolymer. Sutures, meshes for hernia repairing, meshes, skin, scaffold structure or for even skin graft. Polydioxanone, monofilament suture, fixation screws. Screws are made from this polymer. You see, you have seen the photograph of the screws used for bone fixation. Polycaprolactone is a good biomedical polymer, although it is a little expensive. This is used in drug delivery, in vaccine delivery, vehicle, scaffolds for soft and bone tissues. Polytrimethyl carbonate for suture and orthopedic screws, polyhydroxybutyrate, polyhydroxyvalerate, copolymer of polyhydroxybutyrate and polyhydroxyvalerate. These are bacterial polyesters. These are actually bacterial polyesters synthesized by bacteria, bacterial polyesters, bacterial polymers. These can be formed into films, sheets, spheres, fibers as well as bone pins. So, from this list you see large number of applications are there for which we need polymer, and these are the polymers which have been proven to be bioactive, which are proven to be inert, which are proven to be inert in the system, means biocompatible. They are biocompatible, bioacceptable, and sometimes they are biodegradable. Few more, these are hydrolytically degradable polymers, polyurethanes, polyester amide, polyorthoesters, polyanhydrides, polyanhydride coimide, polypropylene fumarate, pseudopolyamino acid, polyalkyl cyanacrylate, polyphosphagens, polyphosphoesters. You see, if you have this literature with you, and today if you see These are here is a list of polymers available for so and so applications. Today you may not, I am not asking that you have to remember everything, no, it is not possible, even for me also it is not possible. But if you see that these are the things available, if it is exposed to you, so from that you have seen here, you see large kind of different applications using these polymers. So, in your professional career you may find some your convenience that time if you need so. Okay. Then hydrolytically degradable polymers as biomaterials, collagen, collagen, this collagen can be harvested from animals. Okay. Natural polyamino acids, synthetic polyamino acids, elastin, polysaccharides of human origin, polysaccharides of non-human origin, and you see their applications all in biomedical applications. Now, these things are today are converted into nano devices, all these things. Now, it is not easy to say in 1 or 2 hour lecture to show the design and process for making those nano devices and their applications. But what I want to say that you can make in nano dimension in order to exploit their unusual properties. This nano devices can provide us some unusual properties which is not available from macro or micro systems, whereas it can be available from these nano systems. There are also applications in tissue engineering. T means tissue engineering, NP means neural probe applications, artificial neural probe for sensing in sensors. I will show you some photographs or diagrams there, and in biosensors also. So, these are the here in all these cases conducting polymers. Now, you see conducting polymers are used in making semiconductor devices. You will be surprised to see that such conducting polymers are used in biomedical purpose also, in biomedical field within heart valve, or making or for some drug delivery, or artificial muscle, actuation of the muscle, movement of the muscle, that is done artificially. Now, if there is some paralysis due to non-functioning or malfunctioning of nerve, okay, that can be activated by these artificial systems using this neural probe system, using this conducting polymers. These are today these are very much used in nanomedicines, nanotherapies. Yes, please.
Sir, we talk so much about drug delivery. Is it reachable to common man basically?
It is there already. It is there. Today, you see chemotherapy. In chemotherapy, this your drug delivery, chemotherapy, drug delivery, this nano systems are used being used today. We do not know, but it is being used.
Everything will be much more costly.
First, I like to live, then I think of cost, because my life is costlier than anything in the world, isn't it? If I like to save your life, should I bother about the cost? I can sell even my country to other country, get the money, and I can save your life. That should be the mode. Okay. So, these conducting polymers, polypyrrole, P dot, I will show you what is P dot, polypyrrole, polyaniline, carbon nanotubes, these are being used for bioactuators, drug delivery. Now, you see conducting polymers are modified chemically and physically to change conductivity. Is it not visible here? So, why you are looking back? I am reading, please listen. Conducting polymers are modified chemically and physically to change conductivity, bioactivity and physical topography geometry. Non-covalent chemical methods like molecular entrapment and adsorption, covalent chemical conjugation and micropatterning using standard lithographic techniques are depicted here. Now, here you see if you take a polymer film, to this polymer film you can attach some bioactive molecules by various means and ways. Now, that attachment can be done through adsorption, physical adsorption of bioactive molecules. So, this is physical absorption of bioactive molecules. This is a kind of modification, overall modification, different types of modifications here. Those things are told, written over here. Okay. This has been taken from a paper. If you are interested, you come to me, I will give you the reference, the review papers, review articles, huge paper, huge review articles, and lots of different references they are given. So, only I have taken few things to explain before you. So, this polymer film, if you want to modify in various ways using these conducting polymers, all these things. Now, that polymer film over which this bioactive molecules can be adsorbed, that means entrapment or fixing by physical adsorption, or you can entrap inside the polymer film, entrapment of small molecules, entrapment of large molecules. You can do micropatterning as it is done in microelectronics. You can modify covalently through primary covalent bonds bioactive molecules. So, if you want to modify your polymer for a suitable application, you take this polymer having certain properties, then you modify it so that it suits properly to that particular biomedical application. This is general theme, this is general concept. Okay. Now, here you see, is it visible from your side? No. This actually photograph is not good. Now, here you see this some fluorescence and corresponding images of neurons on different polypyrrole surfaces. Some neurons you see blue green colors, neurons made of polypyrrole have been developed through some protein molecules, Okay. This is the reference. If it is not that visible, I will give you this PPT, and from there you can see, or if you can get this, if you just download this paper, you can see this there. So, different protein over some protein molecules, these polymers, conducting polymers are deposited. So, conducting polymer deposited protein molecules act as neurons, You take this in a simple way. Protein molecules, where the sequences are there, these are the amino acid sequences, Okay. Over the protein molecule, some conducting polymer, say polypyrrole, is deposited. So, conducting polymer deposited protein acts like a neuron, nerve. You understand? Okay. I am not asking you that just going through or just hearing from me you will understand everything or you will know everything, you will be able to do. No, just focus. I am focusing that it hypothetically you think of some protein molecule over which you deposit some conducting polymer. So, protein is the substrate and you have made that protein molecule conducting like a conducting nerve. Now, here you see this this is a okay, if you feel comfortable you can look backside to that screen. You see this is a polypyrrole based electrically controlled release system. This is the drug loaded system. Achha. In a cultured neuronal network. Now, these arrows, red arrows indicate electrodes. These are the electrodes modified with polypyrrole film loaded with some neurochemical drug CNQX. Okay. So, these electrodes are covered with or deposited with some polymer, conducting polymer, polypyrrole, in which some neurochemical drug is deposited. So, for nerve stimulation, if this device is made, so electrode is there. Now, with the help of electrodes, you can give some, apply some potential, and that application of some potential, I am telling in a crude way, application by applications or through some application of some potential, that conducting polymer will release the drug, neurochemical drug. Whereas, if you swallow that drug or you inject that drug, the therapeutic dose of the drug may not reach the affected site. So, the action will be slow, okay, and will not be proper, but this can help, that it can provide you accurate quantity of drug release from this thing. So, this figure shows this, you see, a specific antagonist of the limit. The CNQX is released directly from electrode. Here these drugs are released and affect cells at the vicinity. Now, these are the neuronal waveforms recorded near a polypyrrole CNQX modified electrode seen before release, after release, before release, after release. Okay. Here the waveform, those waveform has been recorded through this image. Okay. This is very interesting. This is very interesting. Now, I tell you the essence of this thing. Now, here is a polymer, poly 3,4 ethylene dioxythiophene, poly 3,4 ethylene dioxythiophene. This is a famous conducting polymer, poly 3,4 ethylene dioxythiophene. That means a thiophene derivative, conducting polymer of thiophene derivative, okay, poly 3,4 ethylene thioxythiophene. What they have done in this work, they have taken polylactic co-glycolic acid, that means lactate glycolate copolyester. Please listen to me. They have taken lactate glycolate copolyester. It is a biodegradable polymer. Okay. Now, they have made, suppose they have made fiber, very thin fiber, say nano dimension, nano fiber, suppose this PLA, PGA nano fiber they have made, it is biodegradable. Now, to this nano fiber biodegradable drug, they have loaded with some drug. That means, they have made this biodegradable fiber loaded with drug. Okay. After that, they have covered this drug loaded PLGA with P dot, poly 3,4 diethylene diethylene 3,4 ethylene dioxythiophene, 3,4 ethylene dioxythiophene. So, what is there? They have taken a biodegradable polymer, loaded the biodegradable polymer with drug, then they have covered that drug loaded biodegradable polymer with a conducting polymer. Okay. After this conducting polymer in nano dimension, that means you are having a nano tube of P dot, inside which there is a biodegradable polymer loaded with a drug. Once again I repeat, they have made a nanofiber of P dot nanofiber. Within this P dot nanofiber, there is biodegradable PLGA polymer loaded with some drug. Okay. So, you can say this is a nanotube loaded with a drug in a biodegradable polymer. What they did after this conducting polymer nanotube formation, they have degraded this PLGA polymer. Then after degradation of the PLGA polymer, they got this drug loaded conducting polymer nanotube, drug containing conducting polymer nanotube. Within the tube there is drug and tube is made of conducting polymer. So, what is the purpose of taking this biodegradable polymer then? only for drug encapsulation, only for holding the drug or template for the drug or something like that, substrate for the drug. So, they have put the drug in the biodegradable polymer. After putting the drug, after loading the drug, they have covered the drug loaded biodegradable polymer with conducting polymer. Then they have degraded the polymer, where only leaving behind only drug molecule inside the conducting polymer nanotube. Then what they did, they have put this device in the affected site, within the affected site within the body. Then from the external side, externally they have used two electrodes, positive and negative electrodes, and applied voltage potential. So, what they did, on application of the positive voltage, actually it squeezed the tube. On squeezing the tube, drug molecules came out of the system. So, this way drug delivery was there. Clear? These things are described in this slide, particular slide. I think I am clear. Okay. Now, this is again another thing. You see, connector tube. Suppose this is a tube, and this is another tube, this side, two tubes. You want to connect, what we do? You take a connector. Suppose this is the connector. So, we are inserting this part of the tube here. So, this tube is connected to the connector, and if this side is also open like this, imagine this is also open. So, you connect this side with this thing. So, two parts are connected. Now, this is required in surgery, in say bypass surgery. In bypass surgery, what they do, actually if there is some blockade in the artery, the surgeon actually removes that blocked portion, then he takes some that artery or vein from other parts of the body, then they tie by stitching. That is very difficult job. So, this bypass surgery, although there is good success today, but there are failure also, because a blood vessel which is congested due to cholesterol deposition, now that is causing heart blockade, and that blockade is removed by bypass. That means some bypass tube is connected. Now, that bypass tube, blood vessel, vascular tube is taken from other part of the body, and that is connected. How it is connected? That tube should be opened, and this new tube should be connected to that by stitching with the help of sutures. It is easy to think, easy to discuss everything, but how difficult it is to get a success of in this surgery. Now, what they have developed? They have developed a connector made of conducting polymer. What is this? They made a polypyrrole gold bilayer device, polypyrrole over which gold film is there. So, in this bilayer device, this bilayer device in the reduced state is curled. So, here the oxidation reduction phenomena is exploited. In the reduced state, it is curled into roll, that is, then once it is curled into roll, it looks like a tube, and that is considered as a connector, that is inserted halfway into one of the ends of the severed blood vessel. Okay. Now, other half of the device is inserted same way inside the other end of the vessel, and the bilayer expands a few minutes after polypyrrole is oxidized. So, this way, in case of when it is in reduced state, it is curled and formed a tube, then it is put in one side of the vessel, then the other part of the tube is also put inside another part of the vessel, incision vessel, then it is oxidized. On oxidation, it expands. So, it fits to the blood vessel tightly, so without any stitching. So, it connects the two parts of the blood vessels. So, These blood vessel connectors are made from such conducting polymer gold bilayer devices, exploiting their oxidation reduction or their mechanical response by obtained from this oxidation reduction phenomena. Nanomedicine, just I will show you some photographs, I will explain. You read these lines, these are self explanatory. Keeping patience, if you read it you will understand, because I have already described explained the basis of all these things. Nanomedicine based diagnostics, gold nanocells. As diagnostic and therapeutic agents are optically tunable nanoparticles having silica dielectric in the core surrounded by a thin gold shell to provide wavelength dependent scattering and absorption. So, in the diagnostic for the purpose, one can take a silica core which behaves like a dielectric, over which if there is a shell made of gold that can provide scattering, wavelength dependent scattering. So, that can be used in diagnostics or as a probe, say endoscopic probe, USG probe, in order to get the image. So, there is optical property, optical phenomena is required. I do not know the details, I am not expert in this field, but whatever I understand that some electric properties are regulated or optical properties are regulated with the help of these nano devices. Then iron oxide nanocrystals, a contrast agent with supermagnetic properties in MRI for detecting arthritis. If there is some arthritic problem, pain in the bone and the joints, etc., doctors prescribe MRI test. So, atherosclerosis and cancers of breast, lung, prostate and endometrium with high diagnostic accuracy. So, here the nanomaterial is iron oxide nanocrystals. Do you know what is their material, what is the material, what is the device in MRI? We heard this MRI. Now, today we have little information that yes, these are the materials, say gold nanocells or iron oxide nanocrystals are used in MRI test, MRI device. Quantum dots, semiconductor nanocrystals of inorganic elemental core having cadmium and mercury surrounded by a metal shell as fluorescent imaging agents. Okay. This is very interesting. Polymer based nanomedicine for reducing death by cancer. Have a look into this. Actually, this shows some polymer drug conjugates. Polymer drug conjugates. So, this is the polymer molecule which is hydrophilic polymer. These are the drug molecules. So, drug molecules are conjugated to polymer, attached or linked to polymer through some linker. For that, so drug must have some functional site and the polymer must have some functional site. Exploiting the functional site present in drug as well as in polymer, so drug could be conjugated to polymer molecule, and polymer can have either hydrophilic characteristic or hydrophobic characteristics. And again you think of this your emulsion. So, depending on whether this polymer chain hydrophilic or hydrophobic, and the hydrophilicity, hydrophobicity of the drug, conjugated drug molecules, you can make a system of emulsion, nano emulsion. Okay. Now, if this is emulsified, so it is available in the form of fluid. Now, if it is put in the blood vessel, in the blood system, that will be carried through the blood vessel, it will be circulated to the entire body. Now, if there is some affected site, the affected site, there can be have some environment of different pH, different temperature, different surface characteristics at the affected site. What will happen if this system reaches, this linker will be broken, releasing the drug. In other parts of the body, that linker will remain stable, but in the affected site, this linker will be broken and drug will be released. So, this is the concept. Here is in this case polymer drug conjugate. Sometimes proteins are also used as drugs. Some proteins are also used as drug. In that case, that protein can be conjugated to polymer. This is the protein. This is the protein. It is conjugated to polymer. Then entire system in nano dimension can be used as drug, your drug carrier or drug delivery system. Then DNA, sometimes DNA is used as drug. If the DNA can be complexed with polymer, you know what is chemical complexes? What is chemical complex? You know what kind of bonds are present in chemical complexes, you know, complex molecules, you know. So, this DNA molecule can be just encapsulated or somehow enclosed by complexation, all these things. So, that will carry, this polymer system will carry the DNA molecule to some place within the body. Or polymeric micelles, you see polymeric micelles, this is the drug, a cell has been formed by the polymer molecules. So, this system as such can be used for drug delivery. Again dendrimers, what are dendrimers? You know, there is a polymeric system, hyper branched, super branched, supramolecular systems called dendrimers. The core center, it has a large number of branches are produced and leaving behind some spaces inside. Now, that space can be used as template, sometimes can be used as encapsulation by virtue of this your physical absorption of all these things. So, this is system. Passive tumor targeting, please read it, there is no time to read all these things. Passive tumor targeting, slowly. Passive tumor, it is not active, passive tumor. A patient is unaware of this passive tumor. A patient sometimes is unaware of the passive tumor. This is required in diagnostic system. I do not know whether there is a tumor or there is your cancer cells, we do not know, but if it is actually activated or somehow it gives some symptoms, then we go to the doctor and doctor prescribes so many different tests and diagnosis, analysis, all those things, pathological tests. Then only one can come to know whether there is a tumor cells or not, but it remains in passive form. Now, that passive, if there is any passive tumor or not, that can be targeted by virtue of these nano devices. Passive drug targeting, please read it, you will understand. Now, here you see this passive drug targeting through the enhanced permeation and retention EPR effect, enhanced permeation and retention effect. The polymeric nanoparticles preferentially accumulate in solid tumors owing at least in part to leaky tumor vessels and an ineffective lymphatic drainage system. Here you see, this is the tumor site. Now, nanoparticle is injected for passive targeting, locating. Now, what are these nanoparticles? Nanoparticles contain some drug or something, some agent. It will reach the active site only in that affected site. If there is any tumor cell, this will release those agents there. If there is no tumor site, then suppose, for I mean in a crude way I am telling, I am not neither a medical expert on these things. Suppose there is some tumor cell, we do not know how to diagnose. Hypothetically, you see, if something is, some nanoparticle is made into emulsion and that is injected, and if there is some tumor cell, it will be deposited only over there. Then by some imaging system, if it can be detected that yes, that those nanoparticles are deposited at some place, it will only deposit at that place if there is tumor, if there is some cancer cell. If there is no cancer cell, it will not be deposited, it will be excreted through the system. You understand? If there is only affected site, if there is any tumor cell, if there is any cancer cell, all these things, only it will be released and deposited over there. Then outside you have a camera. So, for example, the X-ray, intestinal X-ray, how it is done? So, barium sulphate suspension is swallowed, is not it? What is the practice? You know probably this thing. Then this barium sulphate will be deposited, and this is X-ray opaque, and only that portion, this your some barium sulphate will be, and that you can get the image by X radiation. It is the same kind of thing. This is injected into the system, it will carry, it will circulate through blood. This is nanoparticles, you see, nanoparticles circulated through blood, and if there are tumor cells, it will deposit only over there, it will sit over there. Then through imaging we can detect, yes, it has been deposited there. That means these portions are affected site. Then people will go for, and then they will diagnose, yes, it is a cancerous patient, that tumor is malignant, not benign. Active drug targeting, receptor mediated endocytosis of folate conjugate is very interesting. You see, some conjugate, very interesting, some folate conjugates which are subsequently subjected to membrane invagination. As the endosomal compartment acidifies, the conjugate and the drugs are released from the receptor into the cytosol. You see, this is again a polymer molecule, this is a polymer molecule. Okay. Now, to this polymer molecule, drugs are attached through linker. Suppose these are anti-cancer drugs. So, this anti-cancer drug, if it is circulated through the blood in the form of nano emulsion, then it will be circulated throughout the whole entire body. Now, there might be some affected site having cancer cell. Now, if the cancer cell have some receptor site, then this is identifier actually, like camera or something like that, or what is that called, the security persons today, there is a metal detector. Suppose this is a metal detector, suppose this is a metal detector. So, it is actually after injection, so it will go through the membrane into the cell. Now, if there is only receptor, folate receptor, this is the receptor, this is the folate connected to the polymer to which drug molecules are connected. So, this is a system of polymer and drug containing some folate or some detector like metal detector, and here is a receptor. So, only if this receptor is present somewhere, you see inside is a receptor. These are showing only symbolizing, this looks like this is actually receptor to which this will be attached. Here you see it fits to the size and shape here. So, this is membrane invagination of the system, comes inside, fits to this, and then within this region, if there is some acidity or something like that, here is you see as the endosomal compartment, this is the endosomal compartment. In the endosomal compartment, the conjugate it is acidic or it acidifies. In that acidic environment, the drugs get released. This also detector also release, and ultimately what happens? We get this drug release at the affected site. So, this is a model. This is a model. With the help of this model, this active drug targeting and targeted delivery, how it can be done is shown. So, this is a system containing polymer to which drug is attached through the linker, and it is again connected to a detector, and there will be receptor which will recognize only this, not the others. This receptor, folate receptor, will only recognize the folate, not others. So, if when it flows, when it circulates or moves inside, it will catch. That means, affected site must have the receptor of this folate. If there is, and only if suppose if there is cancer cell, so cancer cell have this folate receptor. If there is cancer cell, it will catch. Then in that environment, this acidic environment, this linker will dissolve, go break, drug molecules will be released. So, drug is actually reached to that specific site, targeted delivery. If we can achieve this success, 100% success, cancer is treatment of cancer or recovery from cancer is not a problem today. Polyplexes. PEG lighted means polyethylene glycol to polyethylene, attachment of some drugs, some polymers to polyethylene glycol. You know, polyethylene glycol is a water soluble polymer, hydrophilic polymer. Polymer is water soluble if there is a limit of molecular weight. Beyond certain limit, it is water insoluble, but it is hydrophilic, okay, because of the presence of oxygen atom. Now, to this polyethylene glycol polymers, some drugs can be attached. Okay. Some drugs can be attached. You see here, you see there are various ways, various strategies of ligand complexes. This polyethylene imine, PEI, polyethylene imine DNA complex. Polyethylene imine DNA complex, if this DNA is used for some therapeutic purpose, so that DNA should be connected through polyethylene imine polymer, again which should be connected to which should be taken help of this polyethylene glycol polymer. So, this is polyethylene glycol to which other polymers are attached or drugs are, these are drugs, you see is polyethylene polyethylene glycol is a ligand, Polyplex. So, Polyplex is a device. Polyplex is a device for drug targeting and delivery. PEG is grafted to polyethylene imine. PEG functional groups are located at the end of the chain for chemical conjugation to ligands. Polyethylene glycol having active sites to which these ligands are bound by chemical reaction, to which DNA molecules are complexed. For DNA complexing of DNA, you have seen in earlier case complexing of DNA, it needs some polymer of some your chemical structure and configuration for DNA complexation. Again, for carrying such polymer DNA complex to the affected site, it is grafted to, that polymer is grafted to polyethylene glycol to make it hydrophilic. You understand? Okay. I understand you are getting impatient. Cellular internalization of drug in polycaprolactone polyethylene oxide micelles. Look at the micelles. These things I have already covered, that much. This is a core cell diagram for drug delivery. Core cell. This is the cell, this is the core. This is the polymer molecules attached to this core. Here is the drug. This is the drug in the core. Thank you.